Once a patient has been identified with a multidrug-resistant gram-negative organism, our options become more limited than once we see a susceptible organism, but we do have options. There are beta-lactam, beta-lactamase inhibitor combinations such as piperacillin and tazobactam that may be active against some of them. That would be active against some of the ESBLs, though there's a debate whether that is a good option for those infections or not. And some resistant Pseudomonas infections, though it's common for Pseudomonas to be resistant to that as well. We have carbapenems that can be utilized. Carbapenems are active, they're kind of our most broad spectrum category of antibiotics, and they're active against some of these resistant species. For example, they're active against ESBL-producing organisms. However, obviously, carbapenem-resistant organisms would be resistant to carbapenems basically by definition. There are polymyxin antibiotics, colistin and polymyxin B. These, in my opinion, are truly terrible options. They're really last-line drugs that we don't use until we absolutely have to, for a couple of reasons. One is that they have significant toxicities. They also have limited pharmacokinetics. Colistin in particular is administered as a prodrug, and that prodrug is eliminated renally, and patients that have, ironically, very good kidney function may eliminate so much of the prodrug that it's hard to get them to a therapeutic concentration. This is not an issue with polymyxin B. However, polymyxin B has pharmacokinetics. While it's not eliminated renally, it's so old that its package insert actually recommends dosing it by renal function, even though it's not eliminated that way. So kind of a significant knowledge gap that we've been working to fix ever since. The aminoglycosides are options. However, aminoglycosides have their own toxicities to them, probably a little bit over-exaggerated compared in people's minds compared to how they really are. However, they also have pharmacokinetic limitations. They're not ideal choices of monotherapy, particularly for infections like pneumonia. And we have some new agents, two cephalosporin and inhibitor combinations, ceftolozine, tazobactam, and ceftazidime avibactam. Both of them are active against resistant Pseudomonas. Ceftazidime avibactam is active against CRE as well and has become a useful agent in those infections. And recently, the drug meropenem-vaborbactam has been approved, which is highly active against CRE infections as well. Recently, there's been a lot of attention to colistin resistance that has been detected originally in China through the MCR1 gene that's been found in basically agricultural samples of E. coli. And since then, it's been found in other places, which is not surprising. Whenever someone finds something bad, it seems everyone else just needs to look, and then they find out that they have some of the same issues. Now, colistin resistance is certainly a significant concern because it's a last-line agent, and if you're resistant to that, what are you going to use? However, I think it's an issue that has been around for a long time, and we just didn't know because we didn't look. It is not typical to test isolates in a hospital for colistin unless you already have an isolate that's resistant to so many other things. So if we start looking back to see how many of those isolates over the last 5 or 10 years are colistin resistant, I think we'd be surprised at just how often we would see that. Many of our agents that we have available for multidrug-resistant infections have limitations. Colistin and polymyxin B have pharmacokinetic limitations, including potentially limited penetration into the lungs. There's some controversy as to whether they achieve sufficient therapeutic concentrations when given intravenously. But there's no argument about how nephrotoxic the agents are. They are significantly nephrotoxic. And unfortunately, we're often giving them to patients that are most vulnerable already, who have been in the hospital for a long period of time, who have significant comorbidities, and these drugs can insult their kidneys that have already been insulted from other things. The aminoglycosides have limitations as well. While not as toxic as the polymyxins, they also do have nephrotoxicity and limited pharmacokinetics penetration into areas such as the lungs. They actually get in there fairly well, but not at high enough concentrations relative to the MIC of the organisms that are often causing infection, most notably KPC-producing organisms and Pseudomonas aeruginosa. Tigecycline is another agent that is available and active against some of these infections, such as ESBL producers and CRE. Tigecycline, unfortunately, has a warning for increased mortality. Relative to its competitors, that increase in mortality was not high, but it was statistically significant, and that has led to a significant decrease in its overall use. And it also has a high incidence of causing nausea and vomiting.